So we're on to carbohydrates now. So this is the third and final one for standard level of the 4 macromolecules. So we've got nucleic acids for high level. They're often ignored, so often it's just regarded as 3 macromolecules. So your body's made up of proteins, lipids and carbohydrates. And so this is the third and final one, and these are the things you need to know. So this is the formula that I don't learn because I wouldn't be able to remember it anyway. My heart would sink if I ever saw that in an exam. I'd have to grab the actual compound from the data booklet and suss out what it is. So it might be helpful to know it's carbons plus waters. It's not that hard to remember really, but I just don't bother. So there are 2 types of monosaccharide, 2 types of carbohydrates. So there's the monosaccharides, which are 1, and then the polymers, which are joining them all together. Okay, so you can have disaccharides, which is 2 as well, just like you can have dipeptides. And so moving on to the next one. So the real focus of this unit is the functional group. So just really applying your topic 10 knowledge. So there's a carbonyl group here, but we really talk about it being an aldehyde and a ketone. All right. And so well, on a further slide, this can be oxidized, you'll see, and this cannot be oxidized, and this one still can be, because you can convert that to a carboxylic acid still, and so this will still be called a reducing sugar, and this will not, because it can't be oxidized anymore. Okay, so cyclical forms. The important thing to note is when we're talking about these things, wherever this linkage here is, which is a glycosidic linkage, again in another slide, in the following slide, we number from this direction here. So we go clockwise, so numbering 1, 2, 3, 4, 5, 6, that's how we number the carbons. You may be asked to go from this to this, and so just draw it out, and just, there has to be this C-O-C bond, so draw a line, and then the leftovers here become an OH group, and so the OH group will hang off that carbon, that first carbon. Okay, so the cyclical forms are intramolecular, because they are joining up, nucleophilic addition. Alright, so that is negative and is attracted to the nucleus there. So there are either 5 or 6 carbon rings between the carbonyl and the hydroxyl groups. So you can see there's 5 or 6 carbons here, I think. Just 6 carbons there. Alright, so in your data booklet you can see 5 and 6 carbons. So you don't need to be able to work out how to go from this Fisher projection to the Haworth projection. That's actually quite difficult. But you do need to know that this is the first carbon, and it goes in this direction here, and you will need to be able to join them up. Just for curiosity, you don't need to know this either, it's in your data booklet. This is the OH facing downwards is the alpha form, and the OH facing upwards is the beta form. So they have different chemical properties and create different types of carbohydrates. So this is something that you could be asked. So draw the cyclical structures. Hopefully they've got it to you in that sense. If they don't, just go to the first, just go to the bottom carbon here, and the one with the OH popping off. And so this will go to here, and this will go to here. That'll get your C-O-C bond, and then you throw an OH coming off the first carbon. So that one has it come up here. So does that work out? There's a group coming off here, this group coming off here, that's correct, here and here, and so that looks correct here. This one's got their C at the So this one here, again, you just draw the line from here, that's the glycosidic link, and there'll be an OH group coming off there. Let's just see if we got that correct. So yeah, there's the OH group coming off there, and there's the glycosidic link. Okay. So as I said before, this can still be oxidized more. So these sugars are reducing agents. There are the conditions. Just a bit too much, really. Could be in the test. There it is. It's all on the PowerPoint. I'm not recommending learning that. If you really need to do that, you wouldn't just Google that, you'd Google a little bit more than that to make sure you're using the latest method or having a look at the different range of methods you could use. So here we have two separate monosaccharides, and you have to work out how to create the glycosidic bond. So basically you find two OH groups so that you can rip out a water from that and still get the ROR bond in there. So that's how you do it. This is just an example of what different sugars look like that are very common. So lactose in milk looks like this. So you can see they're beta types. One's a galactose, one's a glucose. Maltose is also very common. And sucrose is what we just showed you just then. Alright, so this is going through a problem here. So that's sort of just a repeat of what I just did. Deduce the structural formula of sucrose. So you can see these are, there's the two OH groups here. So you're just going to rip out this and then join this to here, basically. And so you can see that that's what happens there. Don't forget to put the H2O on the other side. So this goes to this plus H2O if it's a test question. Okay, so polysaccharides, there's a lot of memorization here as well. So being aware what starch, where it comes from. Cellulose is for cell walls. Glycogen is the energy storage in your muscles. What do they look like? So I have actually seen test questions on this asking what amylose is, which is quite nasty. So 1, 2, 3, 4. So it's a 1 to 4, 1 to 1 to 4, and they're alpha glucose glycosidic linkages. That's what amylose is, if you're going to remember that. You're definitely going for that 7. It's pretty tough. And so this one here is a 1, 2, 3, 4. But at the 6th carbon, there's also a 1 to 6th carbon every 20 to 24 repeats. That's amylopectin. So if you really want to know what starch is, it's a combination of amylose and amylopectin. The amylose has 1 to 4 alpha glucose glycosidic linkages. Amylopectin has 1 to 4, and every 20 to 24 repeats has a 1 to 6 alpha glucose glycosidic linkage. Ouch. Okay. Don't expect me to remember that. I'm sorry. I would memorize that before a test. It being paper 3, I guess you finished your paper 1 and paper 2, and then the next day you got paper 3. So you can spend the night before cramming, and that's probably what you're going to cram. That's one of the many things in this set of videos. Just to let you know, you should know this already. This is the complex that makes the starch turn black when it reacts with iodine. So they might ask you what the test is and what color you see. Hopefully they won't ask you anything nasty like that. I'm pretty sure they won't. So having a look at some other polysaccharides for some other nasty questions. This one looks very much like amylopectin, but the repeats are more common, so 7 to 11 repeats, or 7 to 11 repeats of glucose for the 1 to 6 branching. Cellulose, this is a 1 to 4, but this time it's a beta glucose, and humans can't break this down because we don't have the cellulase enzyme. Okay, all good stuff. I'm sure you're very excited. And what are we doing here? We are trying to, they told you what the glycogen looks like. It's 1, 4 and 1, 6, and you have to work out how to draw it. So 1, 2, 3, 4, 5, 6. So you need to rip off a OH here and create these repeat over here, and then here you need to do the same thing. You need to rip off these bits here with these bits here for the 1 to 6 linkages, and you might even need to put repeats in there. How did we go? You could do even better and do say the number of repeats, so every 7 or so repeats along here, then you get one of these branches coming off. So there's the glycosidic 1 to 6 and 1 to 4 glycosidic linkages. Possibly that's a bit of a nasty one, but that's sort of the thing I'd hope they'd give you in the IB test. Alright, I think this is the last slide. Yes, so dietary fiber, good 1 or 2 mark question they just throw at the end. I'd love to throw the biology question at the end. What's it good for? Dietary fiber moves food through the digestive tract, so it stops constipation and hemorrhoids, fills you up but without giving you the energy, so you don't overeat, so reduces diabetes and obesity, and just the whole fact that it's fiber is and moving the toxins and stuff through, reduces colon cancer, irritable bowel syndrome, and diverculosis. I think I know them more commonly as pulps, colon pulps. I don't know if people use that word very often, so I don't commonly hear that word, but that's alright. Diverticulosis. They're not asking you to pronounce it in the test, just write it. Okay, diverticulosis. And that's it for carbohydrates.